Hello! Today I'll be demonstrating how a DNA molecule is replicated through this magnetic model I've created. I used the back of 2 cookie trays and then cutouts to show the steps so they serve as templates and components that will be working together to replicate this DNA over here. However, I'll only be showing the process from elongation to proofreading. So let me show you this tray over here. This contains the parts that drive the process from elongation to proofreading and terminating. So we've got the ligase, the exonuclease enzyme, the DNA polymerase, as well as the RNA nucleotides. Also on the left, there are all the terms we will use to describe the parts on the left and the processes we can see in the pictures. So here's the DNA molecule. It's got 2 strands. The first step is to replicate it, and then to replicate it would be to separate the strands, and then the next is priming, and in between we would identify the problems that would arise in separation, for which there are other components that work together to avoid this. But suppose we have already done all that, and now we are at this stage called elongation. Also note that to model this DNA, I've just come up with a random combination of the bases in any sequence. So here I made up words for fun, like I did tac, cat, act, GTA, and I repeated it to show a long segment of the strand. Obviously this is not that long, but it's still a little bit long. This would extend a lot, and DNAs are around 1 centimeter long, so they're pretty long. Now, as we know, DNA has 4 bases, which is adenine, thymine, guanine, and cytosine. The A only pairs up with the T, and the C only pairs up with the A. So you can see that over here, they're all complementary. And this is just because of the shape or the geometry of each base that allows hydrogen bonding between the correct pairs. So A would only form these bonds with T, and G only forms with C. Also note that the first one is going to go from 5' to 3', and the second one, the complementary strand, is going to go from 3 to 5'. That means the core of this pattern is going to be opposite: GTA, cat, A to G, and TGA. And over here it looks a little different, and there's a U instead, because in RNA, instead of thymine, there's uracil, and they're fairly the same in terms of structure, but there's a few differences, which is why it's replaced with a U in RNA. So that's why, so A would be with U, T with A, and G with C, and T would remain with A because A is still present in RNA. So after the primase has decided where to start, and the primer has been laid down, the DNA polymerase can do its job. So it will bind to the primer and now add DNA complementary bases to the new chain, which was initiated by the primer from the 5' end to the 3' end, because they are always anti-parallel. So you would pick up the RNA nucleotides, and then you would put it in the position or where we had our primer laid down. So here's the first one, and then there is another one over here, which is ACU. And again, as it works from 5' to 3', we would have another primer set over here, and that's going to be AUG, and there we go. And now we're going to show how the DNA polymerase is going to bind to the primer. So you're just going to take one, going to put it on top, here's the primer, and then on this one, and then on this one. So that's the binding of the DNA polymerase. Now as this continues, more of the strands are exposed, and each time a bit more gets exposed, the primer lays there to allow the DNA polymerase to build. So that's what I've shown here, 5' to 3', but this is just a small segment, so we would continue over here. And this way we get shorter fragments as it's synthesized discontinuously, and this is why this strand over here is called the lagging strand, and the bottom one is called leading strand, as it builds continuously. So let's label that lagging strand. over here at the top, and then leading strand at the bottom. I'm also going to go ahead and just show the hydrogen bonding. So that would be over here, and then the replication fork. This whole thing basically is just, we're looking at one replication fork, and that's it. So when the polymerase is working, it's going to go ahead and add all these new DNA bases and then stop, and then again add all these DNA bases and then stop, and then this way add all these DNA bases and then stop. So these are all added by the DNA polymerase. Notice how the bottom and the top become the same, and this makes sense because it's being replicated. I forgot to do this earlier, but let me also just show you by labeling this RNA primer over here. So it's this and this, and then the... So again, the DNA polymerase could only make the lagging strand in a series of small chunks. And so this primer to primer, this chunk in the middle here is called an Okazaki fragment. So that's what I'm going to show over here. These fragments are normally around 100 to 200 bases long in eukaryotes. Okay, now it's time for editing and kind of getting on to the last step of making the new strand. So because our primer has done its job, another type of polymerase comes in to remove the primers and replaces the RNA nucleotides with DNA nucleotides. So let's just pretend that this is the other kind of polymerase. So it's going to come in and it's going to remove this base. It's going to come in, it's going to remove this space. It's going to come in and it's going to remove this base. So our primers are done working. And now we're taking a look at this step. So as you can see, it's been replaced with the DNA bases now. Now let's look at the next step. It's editing, checking for mistakes. So to show this, the steps to use this model would be then to take this and label. So an enzyme called DNA ligase joins the Okazaki fragments together. So as we were over here, we just run this through. So let's suppose it's joining them all together, and now they have their hydrogen bonds again. So that's the purpose of the ligase. And it does this by the creation of phosphodiester bonds between the 3' hydroxyl group and the 5' phosphate group on the sugar backbone. And so this creates a nice cohesive strand of DNA. So now our editing step is done. Ligase did its job. Now we're going to move on to proofreading. So proofreading, you could just look at the same picture over here. So for proofreading, in case of any errors, either one of those DNA polymerases we used previously, these guys over here, can work as an exonuclease. But I also created an exonuclease to show that. So over here, this E1. So exonuclease is going to cut out, it's going to run through, and it's going to cut out nucleotides that are incorrectly paired at the end of the complementary strand, which is done immediately so that the mistake cannot be copied in the future. So this is DNA ligase. In the same proofreading step, we have our two DNA strands created. So the complementary strand coiled into the double helix shape at the end, so that there's two DNAs each with one strand from the parent molecule, and then our new strand that we just created. And this was the plate or the tray we used with all the components that we need for these three processes. After priming, over here.